more reactive, or supportive. But as you know, solid tumors are very complex. They're all They have blood vessels, they have supporting stroma, and they have a mixture of immune cells that infiltrate and interact with the cancer cells and the tumor cells. And the immune system certainly recognizes cancer and interacts with it. And we'll be looking at this more closely as we go through today's talk. Within the immune system in humans, there's really two components. The first is the innate immune response. And this is not predicated on specific recognition of certain antigens, but is a general response that's mediated through natural killer or NK cells that are present that remove damaged cells or virally infected cells. So this is not antigen specific and is considered the innate immune response. It's thought to be the first response in terms of host response to cancer cells. One component of the natural killer cell response is when it does kill and damage cancer cells may release antigens into the surrounding environment, and these may then become available to antigen-presenting cells that you can see in the center of the slide. These antigen-presenting cells then process the antigens, display them on their surface with major histocompatibility complexes so that inactive T cells can then identify them. With the appropriate association of antigen-presenting cells and inactive T cells can become activated T cells, and these active T cells can sustain a memory function. And in terms of a cancer, this would mean that activated T cells could travel throughout the body and identify and destroy cancer wherever it may occur. So this is part of the adaptive immune response where we acquire immunity to specific new targets. Now, as pathologists, we have observed that the appearance of cancers are variable down the microscope, that some cancers have a lot of immune cells within them, and these are the so-called inflamed cancers. And on the image on the right, you can see that an inflamed cancer may have a mixture of immune cells. They may be effector T cells, or they may be natural killer cells, or even other cells. In non-inflamed cancers, you see tumors that are devoid of cells. They're somewhat deserts of immune cells where there is little immune activity. As we've learned about this, we now understand that cancers that display an immune phenotype may be ones more likely to respond to immune response and may be more predictive of a successful response to immuno-oncology therapy. So this is another role pathologists do play in terms of observing the amount of immune cells within cancers and the amount of tumor-infiltrating lymphocytes, which is now long known to be an indicator of behavior and outcome of many cancers. Now let's talk about the current emerging predictive immuno-oncology biomarkers. We now live in an era of personalized cancer therapy. And what this means is that therapy is now individualized to each cancer patient. To properly individualize therapy, we need to understand both the host and the tumor, and we need the diagnostic tools to be able to analyze those separate items and match them to the most effective therapy. So biomarkers are critical to proper deployment of personalized oncology and proper therapy. And with proper deployment of biomarkers, we will see the best outcomes in terms of cancer outcomes, and will achieve true personalized care. Over the years, numerous biomarkers have been identified and have made their way into clinical use, either as lab-developed tests or as full FDA products. Some of the key biomarkers that pathologists are familiar with include DNA markers, RNA markers, and protein markers. If we look at DNA markers, we've now routinely measured a number of key genes, such as KRAS and EGFR, to look for mutations to identify this correct targeted therapy. EGFR mutations help identify the group of lung cancer patients that will respond to EGFR targeted therapies. There are now new therapies or new DNA markers that are linked to immune therapy response that are under investigation. The ones that are best defined are MSI, or microsatellite instability, that identifies a group of tumors that do respond to certain immune treatments. Others under investigation include TMB, or tumor mutational burden, which may also become an important immuno-oncology biomarker. We're going to talk about that one a little bit later. In terms of RNA is also used as a biomarker. There are many signatures that are in routine clinical use for breast cancer to help classify which breast cancers will benefit more from chemotherapy in addition to endocrine therapy. There's also work now looking at developing RNA signatures to understand the microenvironment in terms of whether it's inflamed or if there's immunosuppression activity in the microenvironment. So RNA will become more important. You can think of RNA as somewhat of a measure of what is being activated within a cancer and also in cells surrounding a cancer. So measurement of RNA can give us information about what cells are associated with cancer, as well as what's going on within the specific tumor cells themselves. Other things that pathologists are more familiar with are actually protein, and pathologists frequently measure protein using immunohistochemistry. There are some key markers involved in precision oncology, such as HER2, that can identify a group of tumors that will respond to targeted therapies. In terms of immuno-oncology, markers that have emerged are PD-L1 and also MMR in terms of markers involved in DNA repair. Markers can vary in terms of their use. So generally, we identify and classify biomarkers into two broad groups, either prognostic or predictive. Prognostic really defines the biology of a disease in terms of what is the arc, the disease trajectory defined by these prognostic biomarkers. Is this an aggressive disease? Is it likely to metastasize? Will it recur? Predictive biomarkers tend to be of greater interest to oncologists in the sense that these allow specific matching of a cancer to a particular therapy. So predictive markers are the ones that are used to select the correct therapy. Immuno-oncology biomarkers are different from traditional genetic driver markers that many oncologists and pathologists are familiar with. On the left, we can see examples of the traditional genetic driver mutations such as EGFR and BRAF, where mutations in these genes can qualify a patient for specific targeted therapy. These are mutations that occur only in the cancer cells. They're not in the surrounding stromal cells. They tend to be stable, so even when the cancer metastasizes, you may still find these so-called founder and driver mutations in the metastatic clones. And often their measurement is binary. It's either present or not, making it easy to select a treatment in this instance. Immuno-oncology biomarkers are different. As we discussed, often they're focused not only on the cancer cells, but on the surrounding immune cells, such as PD-L1, that's expressed in both. They're located on both the cancer cells and the microenvironment. The other thing about them is that they're dynamic and they can change over time. This makes them challenging, difficult to interpret, difficult to measure. And they may also have a fairly broad range of measurement, unlike the binary mutation markers. There may be a spectrum of expression that can make it a little bit more complex to define clear cut points. Now we're going to explore some of the current areas of biomarker research occurring. There are four broad groups, particularly tumor antigens, inflammation in tumors, immune suppression factors that turn off the immune response, and other factors in the host that can modulate how tumors and hosts respond to immune therapy. We'll explore each of these in depth. Starting with tumor antigens, it's now known that many tumors produce something called neoantigens. These are usually altered proteins or antigens that are created often through a process of mutation within a cancer, and that these neoantigens may become targets of immune response. We'll discuss what tumor mutational burden is and how it relates to neoantigens, and also microsatellite instability, this phenotype, and its role in immuno-oncology. As an aside, one should note that not all neoantigens come from mutated DNA, that some neoantigens may be from glycosylation or from post-translational modifications. Those can be more challenging to measure, and we won't really discuss those today. Now, we know that tumors or cancers are often driven by alterations in the DNA, in mutations in the DNA, and that these mutations may give rise to tumor-specific proteins that are so-called neoantigens that can be liberated from a cell when a cell dies or shed from a cell in other ways. When these neoantigens encounter the host immune system, they may encounter at the top right antigen-presenting cells, which may process them and make them available to T cells, or they may actually be presented to T cells by the tumor itself through major histocompatibility complexes on the surface of tumor cells. You can see this at the bottom of this particular diagram, where a major histocompatibility complex is interacting with the T cell. This sets up a cycle of immune activation. So cancer cells that have neoantigens are frequently shedding them. The antigen-presenting cells, those purple cells on the right, are processing them and using those processed antigens to activate T cells. The T cells can then become activated and go back and destroy the tumor cells, continuing the cycle. So in this instance, a very active immune response could be generated. And in fact, in tumors that don't become clinically important, perhaps this happens and they're eliminated. So obviously, tumors that grow and become clinically important are somehow bypassing this in some way. Moving on to tumor mutational burden, you have to remember that tumor mutational burden is a surrogate marker. It's a way to measure the amount of mutations within the tumor genome. As DNA gets damaged nucleotides, appearing as red changes here, accumulate within the DNA, and when a cell cycle occurs, it may be locked into DNA, and that the daughter cells also will now carry these new mutations. And the total number of mutations may accumulate over time. For example, in skin cells that are exposed to carcinogens, such as skin, where sunlight causes frequent mutations, antigens, neo-mutations accumulate over time and can become quite large. The number of mutations can be quite large in these somatic cells. Also, underlying problems in DNA repair machinery may increase the probability that mutations occur and also the probability that they don't get fixed, so that they will then accumulate within the cells, within either normal cells or within cancer cells. So these may lead to the development of basically neoantigens. As abnormal proteins are synthesized, they may be non-functional, they may be secreted by the tumor cell, or as a cancer cell dies, these neoantigens can be liberated into the microenvironment, again triggering the antigen-presenting cells to activate inactive T cells and cause an immune response through activated T cells. Pass over that. Now, through CLAP work, this data is from TCGA, or the Genome Cancer Atlas, and what it's displaying is the frequency of antigens that are identified within various types of cancers that were analyzed. As you go from left to right, they're organized according to the mean number of antigens or the mean number of mutations within the various tumor types. If we look at the bottom, on the far left is pilocytic astrocytoma with the lowest mean number of mutations. If we look to the right, the tumors on the right are melanoma and lung squamous cell cancer that have the highest number of average mutations. These tumors on the right are ones that are presumably carcinogen-induced, melanoma from the sun, lung squamous cancer from chemical carcinogens in the environment. So the other thing to note is that the range of expression is quite large. So that even in the tumors that are far on the right, there are some examples that have relatively low numbers of mutations. And on the tumors on the left, there are some of those that have high numbers of mutations. Also note that the scale is a log scale on the left, so that the distribution is actually much larger than is displayed here. In addition, there are some groups of tumors that you can see in uterine tumors that there are separate pockets that are hypermutated, that there's clusters with very, very high numbers of mutations. And these are the so-called hypermutated tumor phenotypes that probably have underlying mutations in genes such as POLE that are important DNA repair genes. How do we measure tumor mutational burden? So currently there are 3 methods that can be used to measure tumor mutational burden. Typically, it's really dependent on next-generation sequencing. And next-generation sequencing is really the only way one can effectively look at the broad range of mutations that are present in a sample. And as a matter of principle, the larger the number of genes in a panel, probably the more accurate the estimate of tumor mutational burden will be. So we have 3 different ways to do next-generation sequence in general. At the far left are targeted gene panels. So these might be the type of panels that you use on somatic analysis that might be 50 genes or 100 genes or 500 genes that are really focused on identifying the driver mutations and actionable mutations. In the middle is the medium set of genes where whole exome sequencing is done, and about 55,000 human genes are sequenced. This gives her a broader amount of information about whether there are mutations affecting genes. The third is whole genome sequencing, where every piece of DNA is sequenced. One might wonder if this adds additional information to tumor mutational burden, since you are sequencing a lot of non-coding information, and presumably this non-coding information is not generating neoproteins or neoantigens. Tumor mutational burden measurement is still somewhat of an inexact science, and it can give the illusion of accuracy. If you really get into the details of it, one has to be quite clear about where a cut point is, defining, is this a mutation from the background? Is this variant really a true variant, or is this an error of sequencing? So setting cut points and filters are pretty important. The other thing, in formalin, so-called pseudo-mutations can occur as a consequence of formalin fixation. So having a proper pipeline that can clean the analysis and properly identify true mutations from artifacts are important as well. In addition, the genes that are sequenced may play a role. It's thought that genes that are active may be more likely to become mutated than genes that are coiled up and inactive. So the selection of genes in the panel could play a role in terms of what the ultimate mutational burden is. So this is still very much an investigational area. As a consequence, there are numerous groups that are really focusing on this to try to standardize it, and a few of them are listed here. The Quality Assurance Initiative, the International Quality Network for Pathology, and the Friends for Cancer Research are really trying to identify ways to standardize tumor mutational burden measurement and to determine what its significance is in clinical practice. It's important that you understand that the number of mutations in one panel may not correlate exactly with another panel. So if you're comparing, for example, a Foundation panel result to a result from an Illumina TSO 500, the result may not be directly comparable. So it's really important that the pathologist understand how the tumor mutational burden is being calculated for the panel that they're working with, so that they can properly understand what are the cut points and so on for their specific assays. Other genes that contribute to mutations in tumors are the mismatch repair genes, which can lead to mismatch errors in cancers or microsatellite instability. So in tumors that have a deficiency of the mismatch repair enzymes, they're more likely to develop errors in microsatellites and repeat repeat nucleotides of DNA. So after each cycle of replication, slippage can occur where the DNA replicates, and the mismatch repair system repairs these. In cases the mismatch repair system is damaged, these errors that occur in every cycle of replication start accumulating. Some of these errors can affect coding genes, because within coding genes, there may be actually microsatellite regions where dinucleotides are repeated or mononucleotides are repeated as part of the coding region, and where a slippage could change the frame reading and produce a neoprotein, which could lead to neoantigens. So microsatellite instability is a way to look at DNA to determine the amount of damage within the DNA by determining if the microsatellites are stable or unstable. And generally, tumors can be classified as being stable, where there are very little microsatellites are showing irregularities, or unstable or microsatellite high, where more than 2 among 5 standard microsatellites show instability using a standardized panel. Now, if we look at the microsatellite instability high and deficient in mismatch repair phenotype, we see some interesting observations. One is that tumors that have high amounts of microsatellite instability tend to be present at an earlier stage. It's unclear why this biological effect is occurring, but it might be that tumors that have extremely high mutational load because of microsatellite instability may not be as viable, and they may also generate more immune response from the host, limiting their ability to spread. Consequently, this may be why we're seeing a greater representation in terms of earlier stage cancers. We can also look at the relationship between microsatellite instability, tumor mutational burden, and within all cancers that are analyzed. So if you look at the far right, there's a diagram that's got bubbles in it. So out of 60,000 specimens, within those in an analysis, about 4,000 have shown high tumor mutational burden. Within that group of tumors that have high tumor mutational burden, there's a subset that have microsatellite high. So you can see within that group that if you have a tumor that's microsatellite high, it's got a very high probability that will also have a high tumor mutational burden, as these are very interlinked. Interestingly, there is a subset of tumors that do have microsatellite high phenotype that don't actually have a high tumor mutational burden. These are probably worthy of further study, in that the classification of microsatellite high currently relies on only a few markers. Perhaps these are false positives, or perhaps they represent a different type of biology. We can also see in this chart the distribution of various types of microsatellite instability across tumor types, and that we can see the ones that have microsatellite instability high are in tumors that are associated with Lynch syndrome, such as endometrial cancer. Now, there are different ways to measure microsatellite instability and mismatch repair deficiency in tumors. So there are 3 basic general ways that are shown in the columns. The first is using immunohistochemistry. So pathologists can apply immunohistochemistry to look for loss of the key proteins in terms of MLH1, MSH2, MSH6. If you are a pathologist looking at these, you should always be cautious to ensure that the internal normal is there and that those are showing display of the key protein, to be confident that the loss is a real loss that's occurring within the cancer cells. This is a fast way, a fairly efficient way to test for loss of MMR, and is a very powerful tool in the hands of pathologists. One thing to also remember is that when you do identify this phenomenon, it could also indicate that this patient could potentially have an inherited condition such as Lynch syndrome. So the pathologist should be aware of that there is that possibility. The other 2 methods are in the middle, PCR. The PCR method is a standard method that's been around for decades now, where there's a standard set of reference panels. Usually between 5 to 10 microsatellites are analyzed. They're usually run out in a gel or in a sequencer to determine if there's instability at the selected nucleotides. And there's usually a classification system where if one or less are altered, those are considered stable, and if two or more are altered, that they're considered highly unstable. Turnaround time is from 5 to 14 days. And finally, next generation sequencing also sequences across microsatellites. And this opens up a new opportunity in that in some of the panels, there are actually hundreds of loci of microsatellites that can be analyzed. So in comparison to PCR, where there's only 5 or 6, there's 20 or 30 times more microsatellites analyzed in NGS. This opens up a new problem in that we don't exactly know what the correct cut points are for NGS yet to find tumors as high and low using NGS criteria. The other drawback is that it can take 10 to 14 days to get a result. However, with the more increased use of NGS in terms of analyzing tumors, this data will become more commonly available so that we will both have the PSI data from NGS and potentially the tumor mutational burden data as well from NGS. In addition, there may be opportunities for additional bioinformatics on the tumor mutational burden to determine if the antigens are actually in areas that could present antigenicity or if they're in secreted genes and so on. So there's a lot more work to be done as we go forward. Now moving on to immuno-oncology biomarkers and particularly inflamed tumors. So we're going to cover 3 key biomarkers in this area. So these are the programmed death ligand, PD-L1, tumor-infiltrating lymphocytes, and inflammation gene signatures. As we go forward, PDL and PD-L1 have been the subject of intense investigation in terms of their biology and are also important targets in terms of drug pipelines. PDL and PD-L1 are ways that cancer cells interact with T cells. So PD-L1 is expressed on both tumor and immune cells as a way that tumors can stop T cells from attacking them. They become an attractive target in terms of immune therapies, reverse this blockade where tumor cells protect themselves from activated T cells. Now, if we look at the expression and the prevalence of tumor PD-L1 as defined as greater than 1%, we can see the proportion of cancers have a variable number or a proportion that actually have overexpression of PD-L1. At the far left is non-small cell lung cancer and melanoma that have about 60% of the cancers have overexpression of PD-L1. And then at the far right, we have small cell lung cancer and renal cell cancers that have a lower proportion of PD-L1 expression, although it's still expressed in a subset. And it should be noted that the frequency does vary. Now, one of the problems with PD-L1 is that there are multiple ways to measure them. There are multiple clones available, and there are actually multiple cut points. So this is a very, very challenging area for pathologists. So that is a challenge in terms of deploying these assays into pathology labs. There have been some concordant studies that are comparing the various clones. You can see them listed here. There are 5 clones that were analyzed in these concordant studies, 22C3, 28-8, SP142, SP263, and 73-10. In general, there is high concordance amongst the clones, with the exception of SP142. And this clone in general seems to have a slightly different staining profile in that it doesn't have as high a percentage of stainability and also tends to stain immune cells. Although if we do look at the charts, it is a strong correlation with the others, although the results are different. The other area of importance is tumor-infiltrating lymphocytes. So tumor-infiltrating lymphocytes are immune cells that come into the tumor and can kill it or interact with the cancers. So cancers can be characterized by the number of immune cells that are infiltrating, and this may correlate with the degree of inflammation. They can be measured by immunohistochemistry, flow cytometry, and digital PCR. In addition, there's a lot of work now being done on digital image analysis to be able to really quantify these infiltrating cells and to classify them, much like doing flow cytometry in situ. A lot of this work is still exploratory, but it is showing promise, and much of it may eventually get into routine clinical practice. Another type of marker is a gene signature. The way this works is extracting RNA from a cancer or tumor and then measuring the profile various messages within it to get a fingerprint. Using a gene classifier, it's possible to identify tumors that have a so-called inflammation gene signature. And for example, things like interferon gamma gene signature can identify tumors that have activated immune response or inactivated immune response. And these methods are under investigation as additional tools to determine whether a tumor will respond to a selected immunotherapy. These haven't yet made it into routine use, but are under intense investigation. Now, the converse of that is, are there elements of the immune system that turn off immune response? And clearly there are, because biologically, if you have a wound response or a inflammation response, if you didn't turn it off, it would basically probably end up killing the host in out-of-control inflammation. So there is a balance to turn off inflammation. And cancer cells use these biological mechanisms to suppress the host response against them. So there are a variety of biomarkers that may define immunosuppression within the microenvironment. The ones under investigation include lymphocyte activation gene 3, or LAG3, regulatory T cells, or Tregs. The number of them in the immune microenvironment may suppress immune killing of cancer cells. There's a type of suppressor cells called myeloid-derived suppressor cells that can occur within some tumors that can dampen immune response. And there's also an enzyme called IDO1, or indoleamine 2,3 dioxygenase, that can also suppress the immune response. So LAG3 is an immune oncology biomarker that's under investigation. It's a checkpoint receptor that can be expressed on activated cytotoxic T cells that can be measured using immunohistochemistry. LAG3 and the other checkpoint inhibitors, like PD-L1, are co-expressed, and they may be expressed on exhausted T cells. So they could be a good marker that are identifying tumors that have inactive immune responses. Other markers that are under investigation include regulatory T cells. So the content of them within the tumor could be an important biomarker. These can be measured using immunohistochemistry, potentially RNA analysis, or digital image analysis. Also, as discussed, IDO1 is an enzyme that can leak into the intracellular space that affects breakdown of tryptophan. Tryptophan is essential for the survival of certain key immune cells, such as cytotoxic T cells. So tumors that overexpress this will basically starve the immune cells and block their activity. So that's another mechanism to inactivate the immune response. Myeloid-derived suppressor cells are special cells that the cancer can recruit to the tumor microenvironment that can really inhibit the function of other immune cells. So cancers that have a large number of these may be immune-suppressing cancers. More work is now being turned to explore the host environment, and the microbiome is one area under investigation. The microbiome is basically the living microorganisms that are present within our bodies. And a human, it's thought, has as many as 10 microorganisms for every human cell in a person. So there are vast numbers of organisms that live in various niches in our bodies, and these microorganisms may change under different environmental stresses. So there's flora of the gut and oral cavity, and these functions are probably, or these microorganisms, they have an interplay with the immune system and have some sort of balance within the immune system. Alterations in the microenvironment may adjust the immune system or impede the immune system. So there's a lot of active work now in this area to understand the relationship between the microbiome and how the immune system is functioning and how a patient may be responding to therapy. Moving on now to testing practicalities. So there are several general considerations for biomarker testing, and this is an area that pathologists play a really pivotal and key role in. And I think pathologists are an essential part of the immuno-oncology team to help ensure that the proper tissue acquisition occurs and that tissue is handled correctly to enable biomarkers to be managed and measured appropriately. There's a function of steps that start with the tissue acquisition step. Firstly, is the tissue appropriate and sufficient to allow for biomarker testing? The pathologist should also be aware of how the tissue might be potentially used. That sometimes pathologists who are unaware of the additional biomarkers that may be necessary might deplete a specimen by using a small specimen for diagnostic purposes, whereas a diagnostic question is actually not the pivotal issue, and a biomarker question is more important. So this is resolved by having proper communication with the clinical team as to why a specimen is being done and what tests are being considered for that specimen. So the pathologist is critical to ensure that the adequacy is there and that sufficient material is present. Another important step is the pre-analytics in terms of how is that specimen handled. Some biomarkers are more stable than others. DNA biomarkers tend to be quite stable. However, protein biomarkers can easily be degraded. The important aspects are the period of warm ischemia, and ischemic times can lead to rapid degradation of messenger RNA and also to proteins. So it's important that the amount of warm and cold ischemia be kept limited, ideally less than 1 hour, to ensure that the biomarkers are preserved. The other thing is that fixation is keenly important as well. Underfixation is often more important than overfixation. Tumors that experience underfixation may see protein degradation. The ideal window of fixation currently appears to be 6 to 72 hours. It's also important that the specimen is exposed to adequate formalin and that is chopped small enough so that the formalin can actually penetrate and fix the tissue adequately. So these are key factors in terms of getting high quality specimens, and these are areas that pathologists should be highly informed on so that they can be sure that the specimens that come through their labs are in the best shape possible for subsequent biomarker analysis. Creating a standardized process for managing the pre-analytics and tissue acquisition is important, and also creating a timely response for biomarker testing. That one challenge is submitting specimens to outside labs. So if you're working in a smaller lab where materials are being referred to a larger reference lab, it's important that you recognize that time is often of the essence in terms of patients who have advanced cancers who are being qualified for clinical trials. And it would be ideal to expedite the submission of these specimens to the other labs to enable the biomarker analysis to be done so that treatment can be initiated or trials could be ruled in or ruled out as quickly as possible. And ideally, that specimens be released to other labs within 3 days, or within an internal lab within 24 hours would be a standard that would be ideal to be met. There should also be consideration of reflex testing for certain types of assays that can be developed in discussion with the clinical teams to expedite the correct series of testing. Because, for example, limited specimens from lung may have to go through a series of tests to consider not only targeted therapies, but immune therapy. And a proper testing algorithm is essential to ensure that the specimen is handled in as efficient a way as possible, and so that the data can then be made available to the necessary treatment teams as quickly as possible. The other aspect is multidisciplinary communication is absolutely essential. So pathologists play a key role in connecting the oncologists, the radiologists, the surgeons, and providing the biomarker information that's necessary to drive treatment. Ideally, multidisciplinary rounds are a great avenue to share information, but if that's not possible, other mechanisms to interact with the oncologists and care teams are essential to ensure that your lab is performing the key biomarking testing that is essential for the trials and for the treatments that are currently underway in your institution. And this is a great opportunity for pathologists to interact with their clinical colleagues. So precision medicine is really redefining the role of pathology. And I believe pathologists are really central to precision medicine. Pathologists are the custodian of the specimens. So with all of the new treatment options and testing options that are available, it's really imperative that pathologists are knowledgeable about how specimens should be handled, what are the potential downstream tests that could be indicated, how should these be organized and coordinated with the care team. And it really is a very exciting time for pathology. It's really a new golden age for pathology in terms of this central role, in terms of assisting and guiding and working with patient specimens in a very, very critical role. So again, to ensure there's adequate tissue, to ensure the diagnosis is correct, and this is sometimes overlooked as well, that the correct tissue is also very important. Is this really a melanoma or it could be a sarcoma? The role of pathologist is key to proper classification and staging. And obviously, biomarkers are essential. And pathologists are really experts in terms of managing biomarkers and prioritizing the specimen and ensuring that those get done in an efficient and timely way. So clearly, pathologists are key leaders in biomarker testing, and they really play a central and pivotal role in the patient's tissue journey and in the patient's treatment outcomes. The optimal patient-centric care is really a multidisciplinary approach. And as physicians, we always should keep patients in the center of our minds, in that we're doing these tests of the highest quality. Whenever I think of a test, I'm always imagining I'm doing it for a member of my family. How would I handle that specimen to ensure that we're always remembering there's a patient behind every specimen, that often the tests and decisions we make are life-changing and life-altering. So it's really imperative that we handle these specimens with the utmost of care, that we have access to the highest quality standards, and that we continuously work to improve the standards of our labs. In addition, it's essential that we communicate with the other components of the health team to ensure that the whole process is working smoothly and efficiently. So precision medicine really starts with pathology. Pathology was always the basis of medicine. It's the foundation of medicine. And as previous leaders of pathology indicated, your medicine is only as good as your pathology. And that's one reason I was attracted to pathology as a medical student and as a resident. I really felt that in pathology, we got to touch the specimens, We really understood the biology of the disease, and we got the opportunity to do key research and to interact with other key members of the care team. So now more than ever, pathology is important for the future of precision medicine. Pathologists will play a role into the interplay of the immune system and cancer cells, so great area for opportunity. Immuno-oncology biomarkers will support improvements in patient care. And being cognizant of the tissue considerations and multidisciplinary use are essential. And limited tissue, it's essential we use it in as essential a way as possible. So that concludes the discussion and the presentation, and we have time to open for questions. So thank you for your attention, and I hope you found the presentation informative. And again, I'd like to thank BMS for assistance with preparing the slides.
Thank you so much for that dive into a key aspect of immuno-oncology and for sharing your experiences. We've received a few questions from audience members, so we'll try to get through as many as we can in the time remaining. But please be assured that any questions that aren't answered today will be answered via email. Okay, so our first question. At practically every cancer meeting, we hear about new biomarkers that are supposedly predictive of response to target therapy or immunotherapy. What evidence is needed to consider incorporating a new biomarker into routine testing?
A very, very excellent question. And in fact, the universe of biomarkers is exploding, and there's potentially thousands and thousands of them, and they're increasing at a breakneck speed. So this comes down to measuring evidence. And in my opinion, one should really, first of all, look at one evidence, so that if there's clear evidence that a biomarker is essential for selection of a therapy, that it's received FDA approval, and that it's a companion diagnostic, I think there's no question that that's a well-qualified biomarker, and that as pathologists, we need to figure out how to make that biomarker available. The next level is getting into somewhat level 2 evidence, where you're looking at emerging biomarkers and whether these biomarkers are useful to qualify patients for use on a trial. And so this is a next level. And then finally, there's a level 3 where these are of interest but are emerging. So I think a pathologist really needs to be aware of where in the evidence spectrum a biomarker is. It moves from the research point into level 3 evidence, where it looks useful but hasn't yet made the standard for routine use. It then makes it to level 2 evidence, where it may be starting to be used as a qualification factor for clinical trials. And then with level 1 evidence, it's really imperative that that marker is used in the patient setting to enable them to be enrolled properly onto a defined therapy. So there is definitely a life cycle and an arc for biomarker discovery, development, and deployment. And it's really up to the individual pathologist to really understand that and determine when is the right time. But clearly the companion diagnostics are the ones that need to be available and must be presented in a very high quality way.
Okay, great. Wow, thank you for that thorough answer. How do you decide which PD-L1 clone to use for IHC testing, and does the clone you use change? Do we all need to send all of our antibodies for testing?
Yes, according to the FDA guidelines, there are certain clones that are matched to specific therapies. So it's very important that a pathologist knows why is the test being undertaken. And this probably involves a discussion with the ordering oncologist to find that there are different clones available. Although the antibodies do perform in a substantially equivalent way, if you're taking them off an FDA approval, you're then into a new era of LDTs where you'd have to qualify that. So I believe that a pathologist should be familiar with the FDA uses of the various clones and when they're indicated. It's not easy. There are 4 or 5 different clones. They have different indications, and that they're run on different devices. So for the best care, you really need to understand what is the indication, and this really requires a conversation with your oncologist so that you understand, is the correct clone being used? Is the cut point being reported in a way that the clinician can properly utilize that? So I think you have to be very careful when you're using PD-L1 testing and really be clear as to why you're using it and who is using the result, and that they're clear that the correct result is being delivered for proper usage.
Great, thank you. Is there any work being done to evaluate pre-analytical considerations and establish standard guidelines?
Yes, I think there are numerous groups that are investigating this, and it's an important area for pathology because I think this is an area that the pathologist could really contribute significantly to, determining how can we improve the pre-analytics of testing. And I believe it's been an overlooked area, and more data is emerging in the problems with non-reproducibility of things like immunohistochemistry testing. And we've seen disasters, for example, in Canada, where estrogen receptor testing was done incorrectly for over 10 years in one of the provinces, and where we really need to pay better attention to the pre-analytics and produce data to understand how can we define standard operating procedure, how can we run pre-analytics in a defined way and improve testing. So yes, there is work being done, and I think pathologists need to own this work, and it's a huge opportunity for pathologists to contribute. And some biomarkers are more unstable than others. For example, a KRAS mutation, you could probably burn it in boiling oil and it'll be fine. But some of the phosphosignaling markers will decay in milliseconds. So not all biomarkers are the same. And we really need to understand how differences in pre-analytics, like freezing, thawing, warm ischemia, cold ischemia, and so on, can impact it. And there may be things you may be unaware of, that a tumor may be devascularized in a patient, for example, a renal tumor, for an hour, and it's experiencing warm ischemia before you even get it. So there may be information that you need to know what's happening to that cancer within a patient before it even gets to your lab. A very important area of investigation, and we're just starting to scratch the surface.
Thank you for that. So given the faster turnaround time of PCR versus next-generation sequencing, can you comment on the balance of running PCR tests up front to get results sooner versus running NGS panels to obtain more comprehensive results?
Yes. In large academic centers where I've practiced, that was a common practice. So you may have a rapid, for example, if you're dealing with a melanoma, you want to know up front whether there's a BRAF mutation present, so that if you knew it was present, that you'd initiate therapy immediately. So having a rapid test available is key. But if that specimen then didn't pass that rapid test, they would then reflex on to a broader NGS panel that might take a couple of weeks. So having a series of tests sometimes is the best care, so that a patient can have a rapid decision as to whether or not treatment could be immediately initiated or whether they qualify for a trial or not. So I think there is a balance of how do we group tests, what is the sequencing of tests, and I think there's definitely a place for both. Because there's also, ultra-sensitive tests such as digital PCR that can also be used for things like monitoring and treatment response and so on. So I think in the past, some people were thinking NGS will replace everything, but I don't think so. I think there's definitely a key role for targeted PCR-based tests that can enable rapid decision-making in short periods of time and can be done on very poor specimens as well, as their sensitivity can be very high.
Thank you. That makes a lot of sense. So does it make sense to test MSI, considering that most MSIH are included in TMB positive?
Yes, there's a subset where MSI may be high that's not TMB. MSI is a defined companion diagnostic, whereas TMB isn't. So MSI is really the key marker TMB is still a marker in development. So yes, it does make sense to test MSI.
Okay. What are your thoughts on the role of digital pathology using image analysis and machine learning techniques in immuno-oncology and building biomarkers?
I think we're going to see more use of this in the future, and I think pathologists should embrace this technology and that it will improve their efficiency, but also give them ability to do analysis that they were incapable of doing before. So traditional pathologists, you can only look at one or two colors and make a subjective estimate of staining intensity. With digital image analysis, we can move anatomic pathology into a truly quantitative and precision medicine diagnostic field where we can accurately quantify the number of cells, we can measure key targets, we can do multiplex imaging where you could look at 4 markers, you can look at 20 markers, and even with mass cytometry, potentially hundreds of markers. And so with that high degree of multidimensionality, we have to rely on computers and advanced image analysis and artificial intelligence. And I think if pathologists don't embrace this, others will. So I think it's absolutely essential that pathologists get ahead of this and own this technology. I think as pathologists become experts in this field, they will see the value of it in improving safety, improving quality, and also improving the value of a tissue diagnostic, because there's so much information within a tissue specimen that multiplex and digital image analysis can reveal. So I think it's really the future of pathology, and bringing in artificial intelligence, it'll be a tool that pathologists can use to really enhance their practice, make their practice safer, and really make more treatment options available for their patients. So I'd say pathologists should wholeheartedly embrace it.
Thank you. We've got time for one more question. This one is a little bit more involved. So jumping from DNA mutation to neoantigen implies the assumption that the mutations are transcribed, translated, and processed for antigen presentation, which means it seems there could be tumors with high TMB but that aren't immunogenic due to a lack of mutational load expression, or conversely, tumors with low TMB but high immunogenicity due to expression of mutations that may not be on a targeted panel. Are efforts to probe mechanisms downstream of DNA mutation, such as RNA, to count the number of expressed mutations?
This is a very astute question, an excellent question, and absolutely correct, that the tumor mutational burden is an indirect measure. There's no guarantee that those mutations are actually converted into neoantigens, and there's no real evidence or clear measurement that those neoantigens are developing an immune response. So alternative ways to look at immunogenicity, as you mentioned, looking at RNA profile could be one method of doing that. There's also looking at T-cell repertoire in terms of, and I think there's a lot of opportunity in this area, where if you measure the amount of T-cell activity in terms of the number of T-cell receptor clones in the peripheral circulation or within the tumor, you might actually be able to determine, is there immune response to antigens within the tumor? You might actually be able to bioinformatically resolve what those antigens are, and you may be able to monitor in real time how the immune system is responding to antigens within the tumor by measuring the T-cell repertoire. So I think we will see tumor mutational burden has a role, but I think it will be potentially eclipsed by those other, more powerful technologies that really are looking at, is the immune system responding to something? What is it responding to? And also, as a tumor goes through its life cycle, it may generate new antigens. For example, if you treat a tumor with temozolomide, it may cause hypermutation in a tumor and generate new antigens, and the immune response may change over the life cycle of a cancer. So I think this is a very, very interesting area that a huge amount of research is possible in, and that we need to keep our eyes focused on. So yes, very good questions.
Okay, thank you very much for that. I'm afraid we're going to have to leave it there, but again, let me reassure you that any questions that were left unanswered today will be answered after the event by email. So thank you once again to Dr. Mayoko, and thanks to everyone for joining us today. I look forward to seeing you at future webinars. Goodbye.